Thank you for the organizers for giving me the opportunity to talk about our latest work. I'm going to be talking about GraphCompass, which is our computational toolbox for comparing cell spatial organization. Let me maybe clear up a possible misunderstanding straight away. We just received a really wonderful talk about the organization of subcellular structures in cells. And now I'm going to be talking about how those cells organize to form tissues. So yes, my name is Merel. Thank you for the introduction. I'm one of the co-authors of this paper, and I'll be taking you through. So let's start by thinking a little bit about why we might care about spatial organization and why it might be interesting to look into it. So cell spatial organization influences tissue state and influences the kind of communication that can happen in that tissue. And consequently, it's been found that cell spatial organization can impact treatment response and disease outcome. So I just put two examples here on this slide. An example from a paper in Cell from 2020 showing that the organization of the immune TME in colorectal cancer can be linked to clinical outcomes. And a second example from a couple of years later, from 2023, showing that spatial organization can impact immunotherapy response. So we were not involved in these papers, but we did receive multiple requests over the last 2 years from our experimental collaborators asking us if we could help with this type of organization. And so we felt that there was this need from the community for easily accessible, easy to use tools. And this is what we aim to provide with GraphCompass. So how do we go about it? We start by modeling our spatial data as graphs. Can I maybe get a hands up of who has worked with spatial omics data before? Okay, so like a fair number, but not everyone. So what does that data look like? The way that we get it from our collaborators is that we have this matrix where we get for our different cells a quantification of how many RNA transcripts we found for a particular gene in that cell. And the nice thing about spatial omics data is that we also get the spatial locations for those cells or spots, because we have two types of spatial data. We have spatial data that has single cell resolution, and we have spatial data that has spot resolution. And in a spot, we can find, maybe 1 to 10 cells. So it's like multicellular resolution. So what we can do is we build a graph based on this gene expression data and these coordinates, where the nodes of the graph, they're either the cells or they're the spots, and the edges denote spatial proximity. So we're going to connect nodes based on how close they are. We can also give these nodes certain attributes. For example, we can give it, a gene expression vector, or we can assign a cell type label if we have single cell resolution data. This kind of representation of spatial data is not new. Representing it as a graph, this has been done for cell communication studies to find spatially variable genes, and it's a good way to represent the different qualities of spatial omics data. So how do we connect our cells? How do we determine which cells are close and which cells are not? Well, if we have spot-based data, it's pretty easy because we have a grid. We have regularly spaced circles. And so when we create a graph there, we basically just get a grid with like a regular topology. But the question is more so relevant when we look at single cell resolution data. And so we can either say for every cell, I'm going to connect it to its 3 nearest neighbors. Or we say, I'm going to connect a cell to every other cell within, say, 20 micrometers. Then, however, you need to decide what number of neighbors do I want to pick, or what distance do I want to pick. And so when we work with single cell resolution data, we tend to connect it based on triangulation. I'm not going to go into that. I'm going to give you an explanation that will probably make mathematicians raise their eyebrows, but we're going to draw some circles. We have 3 cells on every circle. Those we're going to connect. And what is a nice property is that we do not need to pick parameters. We do not need to do optimization. And in typical circumstances, this solution, this graph we obtain, is unique. Now, we have built our graphs. How are we going to compare them? There have been approaches developed for this purpose, and they tend to be based on clustering. So we built the graph, and then we're going to use something called spatial clustering to obtain these different neighborhoods. The disadvantage of cluster-based approaches is that we need to know what a reasonable number of clusters is. And so how do you determine what is reasonable? Maybe you have prior knowledge about your tissue and about the number of neighborhoods that you might be able to expect for the tissue. But maybe you do not have this prior knowledge. Maybe you do not want to inject that prior knowledge because you're using this tool for discovery. And so then cluster-based approaches may not be the best for the purpose that you're trying to use them for. So with GraphCompass, our goal was to provide these methods that require little to no optimization, that are easy to use, and that provides intuitive visualizations. So I'm going to walk you through some of these methods here. And my goal here is to provide an intuitive explanation. So for more kind of like more mathematically rigorous explanations, I will refer you to the paper, but let me provide you some intuition. If you're interested in the organization of a particular cell type, we can build these cell type graphs, and we can compare them with each other using something called a portrait method. What is a portrait? A portrait of a graph is a snapshot of that graph. We're going to take different characteristics of that graph. How connected are the nodes? How far are the nodes? And we're going to collect all that information in a number. And that number we can use to compare how different these cell type graphs are. We can do this for multiple graphs for our condition and then plot these distributions that help us understand the differences between conditions and how large they are. We can also look at the co-localization of cell types. So there we try to understand, we see these cell types that co-localize. We expect some of this to happen based on chance. And so if we see that they co-localize a lot, what explains this enrichment? What explains why these cell types co-localize? And so we use a GLM for this, and we try to assign the effect to both patient effects, because patients differ from each other, and based on chance, there will be some differences between patients. And we try to explain it as a condition effect as well. So we get that patient effect and the condition effect. And there we can also understand the influence of a condition on how often certain cell types can be found together. Lastly, we can also look at the entire graph to kind of like understand what that looks like, taking into account all nodes, all cell types. And there we use topology method. So again, to kind of like provide a bit more of an intuitive explanation, we're looking at the shapes of those graphs. Imagine I've been given like a bag of nodes and edges, and I've been told you have to rebuild the graph over here. And you start by putting the nodes that are closest together. And so you're growing your data until you have achieved that graph. And the way that that data grows, the way that you do that, that says something about the shape of your graph. And you can compare this between conditions as well. Say something about how the structure of your tissue, how that relates to disease states or to developmental stages. So these are the methods that we implement. We have a Python package for that I will link later. But I think that now is a good time to look at an actual example of this. How do we use it? What kind of questions do we answer? And so I have a quick case study here. And the data that we use is Visium heart data, which we deconvolve to obtain pseudo single cell resolution. And we have data for both healthy individuals and individuals that had an infarction some time ago. And so for that infarction heart tissue, we get data from different sites. We get data from the site where the infarction happened, the ischemic zone, as well as data from further away in the heart, which we call the remote zone. And we want to understand, looking at the organization of that tissue, does the infarction, does it kind of like only have localized impact? So do we only see the effects in the ischemic zone? Or do we see more widespread effects in the rest of the heart as well, even though that tissue was not directly impacted by the infarction? And so I want to look here at the bounding box that I drew, this bounding box over here. What do we see there? We're looking at cardiomyocytes, at heart cells, and this color, the color of the node is going to tell us how different these conditions are. And the size of the node is going to tell us how confident we are that we are right. So looking at the control tissue and the remote zone, so the zone that is, the tissue that is a little bit further away from the ischemic zone, they're different. the remote zone is also different from the ischemic zone. It's in this like intermediate state. It's not quite as impacted as the ischemic zone, but it's also not quite healthy. It's not quite the same as control tissue. We see the same here for the filtration curves. This is a different visualization to show the same result. The remote zone, the organization of the remote zone is in an intermediate stage. And so based on these kind of visualizations, we can say that damage typically originates in one part of the heart, in the ischemic zone, but we can observe that the consequences actually extend beyond this initial site of injury. I also have a little code example here. One of the goals of this package is to make this easy to use. And so for these different visualizations, we have functions that are very similar in how they're built up. So if you work in computational biology with Python and you work with spatial omics data, then probably that data is put in an undata object, as we call it. And so you tell the function, this is my data. These are my patient identifiers. These are my cell types. And these are my conditions. Now build the graph and give me this visualization. That's how we built this package. So I want to come to some of the takeaways. I had a second example, but I want to leave enough time for questions. And I have some things to say about building packages for computational biologists as well as experimental biologists. So just about GraphCompass, you can use it to compare the organization of cells in the tissue and understand how the organization of cells relates to pathological states and developmental stages. It is compatible both with transcriptomics and proteomics data of different resolutions, but spot-based and single-cell resolved data. And we have seen that differences in spatial organization can be correlated with disease states and treatment response. Now, I want a couple of minutes to talk about tooling tips, because I think that as a community, computational biologists are really good at building these creative methods that give you interesting results. But I think that we have a bit of work to do in terms of how accessible we make these tools, how easy to use they are. And so I want to give here a couple of tips for you to use in case you are building software. And the first of those is to use a template, a project template. This is going to help you organize your code, organize your dependencies, and put it into kind of like the right shape to eventually upload it to the Python repository. The template that we like to use in my lab is the hypermodern Python cookie cutter template. There's documentation about that online. There's a GitHub page for that. And once you have this code that you kind of like organize nicely, you can upload it online such that afterwards people can really easily install your codes and your dependencies. You create an environment and they can straight away use it for their own data to make predictions. Of course, it's important that you provide some documentation, but that may not be enough. If you write a lot of codes, even if those functions are nicely documented, I may not necessarily understand in what order I have to put them to get the results that I want. I may want to know how you generated your results. How did you put these functions together? And so for that, you may, for example, want to use Jupyter Notebooks, or as we saw in the last presentation, you can also create a visual dashboard. Lastly, if you put this code on GitHub, you have an issue function. People can leave comments. They can leave questions. Do respond to them. That will help you. Improve that package. And I'm not saying this only because I want these packages, this code, these results to be open and accessible. I say that because it can also be beneficial to you. At least for the TIES Lab, the most cited publication by far is ScanPy, which is our Python package. And so for you, I think it can also be very useful to make this accessible so people actually use your tools and then cite them afterwards. So I want to end just with a thank you, thanking the people in my lab. First of all, my co-first author, Maya, she couldn't be here in Canada because of visa delays, as well as my co-authors. Of course, the TIES Lab for providing good comments. We received comments from other people as well, from early adapters that really helped us shape this and turn this into something that people want to use. And I'm providing here also the QR code for our GitHub repository and for our paper. If you do use this and you have issues or questions, do open an issue. We are very responsive and we'd be happy to help. Thank you.
Thank you for the excellent talk. We have time for a question or two. Please step up to the mics.
Okay, I will start. Maybe I missed it, but how do you deal with replicates for your different conditions?
Yeah, so we do provide ways to the methods taking this as an input. So they take these patient identifiers or batch identifiers. And so different methods deal with this in slightly different ways, but we aggregate this information for the different conditions.
Okay, so you do not use the variance of your replicates to do something different in the graphs?
For every replicate, we build a graph separately.
Ah, okay.
Yeah.
Okay.
Yeah.
Okay. And then okay. Another question is, so you mentioned that mainly to identify recomposition in between conditions.
Mm-hmm.
Do you think you could also use the results to identify spatially variable genes in between conditions?
Let me think about it. This is not what the method is built for. So we do not provide these methods. In the current shape, I don't think we can use it for this, but it is interesting. There are certain methods out there that already do this. I think SpiceMix, for example, does this. We could add that as a functionality, but as of now, this is not a possibility with this toolkit.
Okay, thank you.
Thank you.
One quick question. So in comparing the graphs,
yeah,
one thing I missed is how do you anchor or align the nodes?
Mm-hmm.
Is that defined by gene space?
Yeah, so that's a quick question, and it depends on how this data was originally collected and how this kind of like how the field of view was selected. And so, of course, you can get results that are only there because you selected your samples in a certain way to create these differences from different parts in the tissue. Of course, you're going to see kind of like different spatial organization. And so some of that will come from the sampling side. And maybe if you truly sample randomly, then I think large batch sizes will prevent some of these spurious results. For certain organs, there are common coordinate frameworks, such as for the brain. So there you can indeed anchor it to kind of like a certain point of interest. But it's true that if you sort of maliciously sample to get results, that will, that's of course, that's an issue.
All right, thank you. Let's thank Merel again.